The prostate cancer is the third most commonly diagnosed cancer in men in Australia and the third most common cause of cancer-related deaths in men. Nationally, over 2,400 men die of prostate cancer per year. Although there is 95.5-year survival rates for men diagnosed with localized prostate cancer, this drastically reduced to a median of only 12 to 15 months survival for those with metastatic cancer. This highlights the fact that metastasis is the biomarker for mortality in prostate cancer. The hallmarks of cancer comprise 6 biological capabilities acquired during the multi-step development of human tumors and their propagation. Within the context of epithelial carcinomas such as prostate cancer, it is not the primary tumor that is fatal, but rather it is the metastatic colonization of the tumor in distal tissue and organs. This is why we are putting forward what we believe is a revolutionary novel treatment for slowing and potentially repressing the metastasis of prostate cancer. Trefoil factor 1 is a mucin-associated secreted protein coded by TFF1 gene that is expressed predominantly in the gastrointestinal tract mucosa. Normally, the TFF1 protein is secreted in response to inflammation and promotes cell migration to repair insulted tissue. It is known that repeated epithelial insult is linked to the chronic inflammation and unabated trophic effect on the prostate gland, where prostatic tumor cells initiates and thriving. The TFF1 presence increases in this inflammatory environment, and although initially secreted for repair, these proteins can be hijacked to detrimentally induce aggressive cell invasion and migration. Studies have shown that TFF1 overexpression is linked to the downregulation of E-cadherin in prostate cancer cells. This is postulated to be the major factor that leads to tumor invasion and migration, which is a hallmark of cancer. E-cadherin is one of the most important transmembrane protein and is the classical feature of the epithelial cells that functions to provide tight cell-to-cell adhesion at the adherence junctions. Its functional significance makes it a major barrier that tumors need to circumvent to achieve epithelial mesenchymal transition in order to escape from their local environment and begin metastases. Therefore, TFF1 overexpression is pivotal in epithelial to mesenchymal transition because it decreases the expression of E-cadherin.
Now I will tell you why TFF1 is overexpressed. Basically, CPG islands in the promoter region are hypomethylated, causing overexpression of TFF1. This hypomethylation of the TFF1 gene is due to the inactivation of microRNAs. In normal cells, microRNAs are used for gene silencing by their action on DNA methyltransferases, also known as DNMTs. The DNMTs hypermethylate the promoter region, thus silencing the gene. However, the inactivation of microRNAs in prostate tumor cells prevents DNMTs from methylating downstream genes. This therefore leads to hypomethylation of TFF1, which significantly increases its expression. Now we know why TFF1 is overexpressed, then how does this overexpression affect epithelial mesenchymal transition? Let's go back to the E-cadherin, which is constantly expressed in normal epithelial cells. When the TFF1 gene is overexpressed, the resulting TFF1 protein acts in an autocrine and paracrine manner on the tumor cells. TFF1 is dependent on the epidermal growth factor receptor signaling pathway, and it is through this pathway that the presence of E-cadherin is both lost and also degraded. In this degradation process, beta-catenin is released, which further drives the epithelial mesenchymal transition. Now I will tell you how we will target TFF1 overexpression. In order to downregulate the TFF1 gene, we will use antisense therapy. Antisense RNA, complementary to the TFF1 mRNA gene sequence, will be incorporated into a nanoparticle vector and administered via the rectal route to the prostate. The prostate-specific membrane antigen is overexpressed on the surface of prostate cancer cells, and prostate-specific membrane antigen aptamers have a high affinity to these antigens. Therefore, aptamer-modified nanoparticles will be used to achieve specific targeting to prostate cancer cells. Once inside the nucleus of the prostate cancer cells, the antisense RNA will bind to the TFF1 mRNA, causing destabilization of the mRNA, preventing it from being scanned by ribosomes. Therefore, the formation of the antisense RNA TFF1 mRNA hybrid leads to the premature termination of transcription, which causes downregulation of TFF1 protein.